answers which you were supposed to write. Please take note that this is not the final discussed memo. Yes. So the final discussed memo will be released soon. All right, let's go and then find out what we expected to get in this memorandum. What we so let me know if you can hear me properly. If you have any question, let's discuss about what you have maybe challenges with, so that when it comes to the final exam, you don't miss it out. All right, let's start without wasting time. You know me, I don't go through instructions. You're supposed to do that every time you go through the paper. M.SAIDI as usual. The first question is saying that various option chart. The diagram below shows the allele. When you say allele, it means this. Allele for the head in the flowering plant, where the tall is dominant and then small t is short, meaning it's recessive. So once you have a capital letter and a small letter, what I told you is that is complete dominancy. But because there are two different types of alleles, there are two different kinds, not types of alleles, so it means that is heterozygous. So now we come here and then we find out that the plant is homozygous. No, the plant is heterozygous for the height. So that looks to be the most correct answer, which is B. So you get 2 marks there. Then you go to the next question, that which one of the following is not. The key point is not applicable for crossing over. So if you look at what is not applicable for crossing over, genetic material is exchanged between non-identical chromatids of homologous chromosomes. Cool. Homologous chromosomes undergo crossing over. In they're saying not applicable for crossing over. Homologous chromosomes undergo crossing over in prophase 2. So we don't have homologous chromosomes in prophase 2. Therefore, and crossing over doesn't take place in prophase 2. Therefore, B becomes the most correct answer, because the key point was saying which is not. Then we go to the next question. They are saying that which changes occur in the sex of the cell, in the gene of the sex cells. These changes lead to changes in the parent phenotypes. No, what happens in the sex cells in terms of genes does not change the genotype of the parent, so this is wrong. Only affect asexually reproducing. No, doesn't affect, because they don't have even the sex cells. Always harmful. Not really can be a basis for evolutionary change. So why? Because this will lead to variation. So D becomes the most correct answer. Then we go to the next question. They're saying that which one of the diagram below represents the different 4 different species of bacteria. Then they're saying that which statement is correctly regarding the survival chance of this species if there is a change in the environment. If there is a change in the environment, so species have the best chance of survival because it has the most variation. That's what they are saying at A. It could be true, but let's find out if there is the most correct answer. Species C has the best chance of survival because it has no gene mutation. If there's no gene mutation, then automatically it can't survive. That is too wrong. Neither B nor D will survive because they compete for the same resources. It can't be. They're saying neither B nor D will survive. Neither B nor D will survive because they compete for the same resources. What shows that they compete for the same resources? So it makes it none of these species will survive because bacteria reproduce asexually. Say no, 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 not really. So the answer, the most correct answer becomes A. So A becomes the most correct answer. So if you have any challenge, you let me know, and then I find out that this is power. The paper was so beautiful. Majority of the things you did on Saturday. Yes, boy, B boy. Yes, I told you that 90% of our discussion is always the paper. So always stick on our channel, you will get distinctions always. And I will salute that boy from Limpopo. I just want to take this opportunity to say thank you so much for my boy from Limpopo who got 140 out of 150. So that's a good. And however, they robbed you with those 3 marks. 3 marks. Their memo, they had some issues of percentage increase. The formula they gave in the memorandum was not correct, so we claim for those 3 marks. We need them. We need those 3 marks. Yes, I will try to see that that is rectified. All right. They're saying that the relationship between gene and the allele is okay. The relationship between gene and the allele, gene are different form of the same allele. Gene are different forms of the same allele. Each gene are different forms of the same allele. This is wrong. Each person contains two alleles for the gene of one characteristic. Yes. However much the character can be multiple allele, can be controlled by different alleles, but this one, each person is supposed to have two alleles of that character. You understand? Like blood group. You can't be blood group, allele for blood group A and B and then O. Never. You have either A, homozygous, heterozygous, like that. Or if you have for A and B, then it's one allele and one allele, meaning there are two of them. However much there are 3 alleles controlling a blood group, but you only have to be with 2 alleles for one character. That is the key point, and then B looks to be correct. I'm not saying that's the most correct answer. Gene can only have 2 possible alleles. Not related, because we have multiple allele. Because of this word only makes it to be wrong. Allele are phenotypic expression of gene. No, actually this is like misleading. Why? Because it's the gene which determines the phenotype, not the phenotype determining the gene. So B becomes the most correct answer. 1.6, this is a structure which show that there is a divergent evolution. And these structures are called homologous structures. Those people who are using the distinction material book, I think you are very well conversant with this. Study the diagram and the answer and state the structure below. All right, structure are homologous. Yes, the structure are homologous. This is correct. Animals do not share the no. They share a common ancestor. That's why it is divergent evolution. Forelimb of all these animals have the same function. Not really, they don't have the same function, because this is for flying, this is for swimming, this is for maybe making holes and catching rats, and then this is for carrying. So they don't have the same functions. Structure arose due to modification by descent. That's exactly. If you check there, let me show you what I'm saying. Maybe you don't understand what I'm saying here. This is what I'm saying from our distinction material book. This is it. That's exactly, I think if you look at that diagram, it's exactly the same as that diagram. You see, they brought it human, cat, whale, and bat. Human, cat, whale, and cat. I told you, read the distinction material book. Distinction is guaranteed. All right, let's continue and then see. So, and then here we're saying that this is check modification. This is what they are saying, modification by descent. So this is what they are trying to say, that modification by descent. So 1 and 4 becomes the most correct answer. So I have to look for 1 and 4. 1 and 4. 1 is this, and so C becomes the most correct answer. Who got it right? Yeah, who got it? That one right? Who got that one right? Okay, guys, let's continue. All right, they're saying that a certain disorder is caused by a dominant Dominant allele. I emphasized about this dominant allele. The following combination should be considered. Okay, this and this. Okay, they're saying which one of the following combination arose crosses can result in offspring without a disorder. Certain disorder is caused by dominant allele. So meaning that in this case, we are looking for something which has no. Certain disorder is caused by a dominant allele. So where I have capital F, that isn't it. So when I cross this and this, all of them, some will be like this and some will be like this. So which one of the following combination can result in some offsprings? You see, this is the keyword. Some offspring without, meaning that they are recessive like this. So number one is correct. When I cross this and this, I'm going to produce all of them like that. So number two is wrong. When I cross this and this, heterozygous, if this combine with this, I'll form that and that. So meaning that 3 is also correct. If I cross this and this, I don't have any homozygous recessive. So it becomes 1 and 3 and 3. Meaning that I look for 1 and 3. 1 and 3 becomes A. So A becomes the most correct answer. So if you got it right, you need to clap for yourself. Ne? Yes. Hey. Did I say one? Eh? Let me read it again. Someone is saying something. So we are saying that yes, it is one. Let me see. One y1 let me check and see. Yes, well, let me clean here. Y1, because if one recessive allele, my boy, and one recessive allele combine, they form that. And then the 3, if this recessive allele combines with that recessive allele, they form that. And because they say it's caused by dominant allele, automatically it can't be. So it's gonna be 1 and 3, which is 1 and 3 only. So that one becomes the most correct answer, which is A. So I think I'm clear there. Those who didn't understand that concept. We go to 1.8. 1.8, they're saying that approximate. They're saying that they're saying that 1.8 and 1.9 are based on the diagram below. Okay. Approximate how long did the Paranthropus pan existed? How long this pan existed compared to its closest ancestor? So it means that how long? How long did this exist? So this is the pan. So it existed up to from there. From there up to there. So what we need to do is we need to subtract the recent ancestor. Where is the recent ancestor? They're saying compared to the closest relative. So who is the closest relative? So if you look at it, this was the relative it had. So it means that we have to subtract this from that, and then we get this minus this, and then we get remaining with that, meaning that that's how much it existed from that more than that point. So based on that, if you subtract, I saw if you drew them correctly, I saw that B was the most correct answer. But you need to use the paper with the ruler, and then you measure properly. Yes. So I'll leave that as question marks. if you measure properly. Yes. So if you measure properly, you need to use a ruler. Yes. So that you see exactly if it is pointing here or it is pointing in the 5. Yes. It is pointing in the middle or it's a little bit down. Yes. So your paper will show you exactly what is. But when I measured from the screen, so that what it gave me. So they're saying that how many genera are represented in the diagram above. How many genera are represented in the diagram above? So we say that genera, based on genera, we told you that yesterday that genera, you look at one, that's one genus. You don't repeat this one again. So it's the same thing. So it's 1, it's 2, it's 2, is 3, yeah, it's 4. I'm seeing Pan 1, Paranthropus 2, Australopithecus 3, and the Homo 4. Do we have a 4 there? Yes. So C becomes the most correct answer. Let me check and see if you have any challenge there. Yes, no challenge, and then we continue. All right. So, I think, guys, you got 18 out of 18 there. Yes. So let's look at 18 out of 18. Yes. So they're saying that, oh, who got at least 14 out of 18? Yeah, because if you got 14 out of 18, you are still in lie. You might still lie in the same distinction area. Terminologies. Allele that does not influence the phenotype of inheritance. We talked about this. It is recessive allele. It only expresses itself in the presence of a dominant allele. Simple. They are saying that the bond that form between the amino acids. We talked about these bonds so many times. We call it a peptide bond. If you wrote polypeptide bond, 0. Portion of the chromosome that code for a particular characteristic. We say this is a what? It's a gene. Biotechnology process that produce genetically identical. We say it is cloning. Yes, we talked about it when you're talking about the cloning of the dolly sheep. They're saying that the type of variation where there is a range of intermediate. We said it, and we say that it is a continuous variation. And I told you, yes, I told you that anything which you are born with is called what? Continuous variation, because it has a range of characters. That's what I say. And they brought it here that they have a range of characters, or what you call the intermediate. Yes. Let me show you what I'm saying. This is what I'm saying. Yes, this is what I'm saying, and this is the example I used when I was talking. That's what they are talking about. And they're saying that the study of past present day distribution of living organisms across the continent. Yes, living organism is bio, then distribution is geography, so it is biogeography. So that's when it's done. They're saying the scientists who argue that acquired characteristics obtained by the parent could be passed to the next generation or offsprings. Who is that scientist? And that is the Jean Baptiste de Lamarck, or Lamarck. Even if you say Lamarck, I think they will give you a mark in that regard. Hey, yes, we continue. Example I use. We continue to the next one. Yes, Danko, Citro. Hey, I don't know how you read your name. Yes, guys. I hope you got 7 out of 7 there. 7 out of 7 there. Then we go to both A and B. Yes, characteristics of African ape. Prognanthus. Yes, they are prognanthus. They have protruding jaws. So they're saying less pronounced brow ridge. No, they have pronounced brow ridges. So the answer is A only. The word only must be there. Characteristics of blood group inheritance. So what are they saying here? Characteristics of blood group inheritance. Multiple alleles. Yes, it is multiple alleles. And then not all the blood groups are codominants. The characteristics of blood group, they're not saying blood groups, meaning that it could be that one blood group. So both A and B makes to be the most correct answer. Yes. Why? Because blood group A, B is co-dominancy. However, blood group A as A is not codominant. It is complete dominant. You see? So it is codominant, but is caused by multiple allele because there are three alleles which control blood group. So both A and B, both A and B becomes the most correct answer in that regard. Then they're saying that in a cross between the bull, remember it's a male cow. I don't know whether I'm saying it right. A male more right, is heterozygous for long eyelashes. Long heterozygous. The moment you say heterozygous and brown coat, it means that long eyelashes and brown color are both dominant. So meaning that, let me say that eyelashes, I'm gonna say it's capital E, and then color I'm gonna say is capital B, for example. But because it is heterozygous, then it's gonna be capital E, small e, capital B, small b, without a space. They're saying that the cow has short and yes, the cow has short eyelashes and white. It means that those are recessive. So I expect them to be like this and to be like that. They're saying that the gamete of the cow will be the gamete of the cow. In this case, they are looking for the gamete of the cow, not the bull. If they're saying the gamete of the bull, definitely it would have been this. So since it's this and that, so it's gonna be E, it's gonna EB. All of them are going to be EB. EB, isn't it? So because now here's the cow is not having any dominant character, therefore this one becomes wrong also, and this one is wrong already, so answer becomes none. So here it becomes none, here becomes both A and B, and then this one becomes A only. Is it clear? I think, guys, I hope you got also 6 out of 6. So I expected you to get a lot of marks. I think, guys, you are counting and you're also checking and see that things are moving well. Guys, let me know. Let me know what if you have any concern of what we have discussed now. Don't wait when I'm offline and you start to say, sir, now what? Say, how will I say now, sir, when I've already gone offline? It's now downloadable. You understand? Yes. But now I'm on it. Like I'm live. It's not that some of you think I'm not live. Let me show you. Yeah. Yes, you see, I'm live. Ne? So let me know if you have a challenge. Yeah, let me know that so that I'm able to answer those questions, or so that you don't repeat the same thing when it comes to the final exam. You understand? That's why I do these memos and then we discuss about them before even they release the final memo. Why? Because I want you, it's like it's me and you writing the same paper. How will you obtain those marks? You understand? So this, if I discover there is a small challenge, definitely that challenge you'll also have it. So we present what you try to make them to be, you're able to pass if that the same questions come back again. Because once they give you a memo, you just look for where you got it right and you won't get enough time to go through it. Anyway, let's continue, and then but I just want to show you that let me know if you have a question so that I'm able to help you out, and then we solve this problem, and then let's go. Hey. Out of Africa hypothesis. This one we talked about it, ne? But now this time What they did is they stated it for you. They say that out of Africa hypothesis, modern human, you see, I told you the word modern is very important. Next time when they ask you to state the out of Africa hypothesis, you know how to state it. Modern humans originated from Africa and then migrate out of to other continents. So some of the fossil of genus Australopithecus has been discovered in South Africa. I told you don't go without knowing two fossils from at least Australopithecus africanus in South Africa. So they brought Australopithecus africanus, not only Australopithecus, but this one is Australopithecus africanus. Mrs. Place. So give the name of the scientist who discovered that. Who discovered Mrs. Place? It is Robert Broom. And then who discovered the Littlefoot is Ron Clark. So you guys, I had summarized this in your distinction material book. Ne? Yes, I summarized it here. You see, Littlefoot, Ron Clark, you see, Mrs. Place, Robert, you see, they just brought the way it is here from the distinction material book and then they put it, that's it. If they don't ask you this, these are the only things they're going to ask you. If they don't ask you this, ask you this. If they don't ask you this, they ask you this. You just need to memorize this small table, done. However much other books are having a lot of issues. Anyway, let's continue. State this fossil site where Littlefoot was discovered. Fontaine. So you say Fontaine caves. Yes. So I don't know whether if you don't add the word caves, they will give it to you, but Fontaine. Fontaine is Fontaine. All right. Give the species name of fossil, Australopithecus sediba. Yeah, they are looking for Australopithecus sediba, which we call the transitional fossil. Yes. State the fossil site. Okay, we are done with this. Name the genetic evidence support out of. I told you that there are three evidences. I told you there are three evidences. Number one, fossil evidence. Number two, genetic evidence. Number three, cultural evidence. And I say that when they give you cultural evidence, you say examples are tools. When they give you genetic evidence, then you say the example is mitochondrial DNA. We don't use Y chromosome anymore. You understand? So if you wrote Y chromosome, 0. So genetic evidence, which is the mitochondrial DNA. When they ask you fossils, obviously you know the fossils. So you give the what? The example of the fossils. I think I'm clear, and it's clear in that regard. Simple. All right. So we go to that mark. We say the genetic evidence is mitochondrial DNA. Someone can write in the abbreviation. Others can write it here. Sir, please explain the last question. Now you are saying last question. Which question are you talking about? Sir, where can I find this book? Yes, from our link of if you just check there, you say join, you'll be able to access the book. All right. Let's continue to the next question. That book was called Distinction Material Book with a reason, and every year I update. So don't use the last year's version. You have to use, if you are going to do next year, I'm just talking about next year, use the next year's version. You understand? Because every year is updated. Yes. They're saying the diagram below represents the DNA replication. We did this several times. So they're saying during which phase in the cell cycle does DNA replication takes place? So what they did is those questions I told you yesterday is what exactly they brought here. Yes. So during which phase cell cycle is during interphase. They're saying that natural shape of DNA. This one I emphasize, they say that we call it double helix, not double helical, not helical, not coiled. No, the only statement we accept is double helix, because when this guy who James Watson and Francis Crick, they described it as, check, they said that is DNA is double helix. That's the only with complementary base pairs. It's double helix. That's how James Watson described it, and that's how we're supposed to describe it. So I think I'm clear there. Let me check and see this person who said which question. Safia Muhammad. Safia Muhammad. Okay, I think I answered you. Ntime. I don't know which question you are telling me to go through or to explain better, unless you point it out. They're saying that give the letter and the name of okay, sharp. State that we are done with this. Double helix. That's the answer. Name letter and the name of for each of the following monomers. We highlighted this several times. We said is a it's a nucleotide. Even I showed you and I said that guys, please make sure that I said that nucleotide, and I said sometimes they call it building blocks, sometimes they call it monomers. That's what I said yesterday. Yes. So I think you got it right. It is nucleotide. You see, they said building block and then monomer. Exactly how it is written here in the, you see, building blocks, and then they say monomer. Are you seeing? Yes. I think now you are getting it right. Please, you want a distinction? Distinction material book by M.SAIDI. Alright, let's continue. Sugar in this one is obviously it's deoxyribose sugar, complementary base, nitrogenous base, okay. In molecule D, when you look at molecule D, it is you have thymine, definitely is gonna be what is they're saying the complementary base, okay, of D. So they are not saying that in RNA what they just want in the DNA, which is the adenine. So you have to write the word adenine. So someone is gonna ask me, what about when I wrote capital A? I hope they will give you it right. Yes, capital A, they will give it to you. Then we go to the next question, 1.6, so that we wind up with the first section. Identify which of the diagram A, B, or both A and B belongs to Homo sapiens. Obviously, this is Homo sapien diagram A. So you write diagram A. Hominids. We are hominids. Chimpanzees are hominids. Gorillas are hominids. So the answer is both. I'm sorry if you didn't get it right, both A and B. Yes, so the answer is both A and B. Then describe 3 characteristics of organism skeleton that allow for bipedalism. That's exactly the same way we discussed it and highlighted it and I emphasized it. I don't know which language I'm supposed to use here, but this is it. They are here. Foramen magnum, the position of foramen magnum, forward position. Pelvic girdle, short and wider. And then they're saying that spine is S shape. So how many marks are there? They're saying describe. They're not saying explain. Foramen magnum in forward position because they're saying on the skeleton. Foramen magnum in forward position to allow the spine to connect vertically. Now you are explaining. Foramen magnum in forward position. But if you explained it, it's still correct. It's still fine. They will get your marks. Foramen magnum in forward position to allow the spine to connect vertically. Cool. Get your marks, né? Yes. They can't cross it because you added something which sense. You understand? Yes. So they're saying that foramen magnum forward position, okay, to allow the spine connect vertically. Then number 2 is the shape of the spine, which is S shape to allow flexibility. Yes, and then pelvic girdle short and wider to support the upper body weight. So exactly that's how you were supposed to write these 3 points bullets. And you see I put them in different colors, which shows that those are different marking points. Yes, these are different marking points. Oh yeah, man. You did not mention later. I did not mention 1.53. 1.5. 1.5. Oh, thank you. Thank you. Thank you. You see, that's how 1.5, 1.53. You said I did not mention the 1.53. Oh, thank you so much. Monomer, thank you so much. Thank you so much. Nkanyisire. You see, that's the reason. That's why I told you that sometimes I'm rushing. And when I rush, I end up forgetting some things, you see. So, yes, Nkanyisire, thank you so much. They're saying that 1.53, it is letter. Monomer is D. So, it is D, which is a nucleotide. And then sugar of DNA, we said is deoxyribose sugar, which is C. And then the complementary base for nitrogenous base found in D. Found in D, which is adenine, and then in this case it is A. They just said it. So you guys have to say adenine because A is adenine. And then this, because this one was obtained from this side, you understand? It was obtained from this. You see, the arrow is showing it was obtained from this side, therefore adenine from this side. So that is it. Thank you so much. Thank you so much. Raswell, you will get it. Don't stress about it. Multiplication were tricky. Okay, thank you so much, guys. I think section A, state 3 characteristics of the scar that allow binocular. Here, the characteristics of the scar that allows binocular, the eye sockets, they are in front, eyes in front. You understand? It has two eye sockets. Basically, if you have one eye socket, because they're talking about the characteristic of this organism, to allow what? Binocular. So two eye socket. So if you say eye socket, two eye sockets, they're in front, you see, so that they're able to see. Binocular means you're able to see with the two eyes. So two eye sockets meaning that they will have two eyes. You understand? Yes. Think that is it. If you got another answer which is correct, then they'll also consider it based on the discussion of the memorandum. Who got 50 out of 50? Who got 40 out of 50? And if you got at least 40 out of 50, yeah, you a star. And then if you got 45 out of 50, you a moon. And then if you got 50 out of 50, you are a sun. Yes, all right. Question 2. Let's go to question number 2. Hey. La. The table 1 shows the partial DNA sequence of human, and table 2 shows the amino acid coded for by different messenger RNA codons. Best triplet number 1, 2, 3, and 4 human DNA molecule. Right. Name and describe the process in protein synthesis that is responsible. I told you this. Formation of messenger, that is what that is the transcription. But I told you that if you mention the process, they give you a mark for that. So so it is here, transcription, formation of messenger. So when you are describing it, you describe it. You understand? You describe all of it. Transcription means formation of messenger. So they say the name. So you mentioning transcription is a mark. So mentioning. So in description, you have to get at least you have to get 4 marks. I released a simple video which was playing nice, and just to make sure that you remember this, you don't go to the paper without knowing it. I hope you guys, you listen to it and then you're able to get a distinction, or you get able to get good marks in this regard. So that is done. I'm not going to go through, but double helix DNA unwinds, weak hydrogen bonds break, double helix DNA unwind. You understand? Weak hydrogen bonds break. You understand? Then one strand is used as a template. So here I used a different version. So here it forms to form two separate strands. One is acting as a template. So you have to describe it. They just want only 4 points from there, and then you get it right. All right, describe, give the messenger RNA codon that is formed from the base triplet number 4. So base triplet number 4, I come here, I look for base triplet number 4. So they're saying because this is DNA, so a messenger is gonna be U, G, U. So here is gonna be G, U, U. So for that answer, they are saying name the amino acid coded for triplet number 2. When I come here, I need to find out what is here. It is messenger RNA. I explained this, and then so it means that I just have to convert it one time. So if this is gonna be UGC, UGC, UG, where is it? UGC. It's here, which is cysteine. Yes, so the answer there is going to be cysteine. So this is the answer for that. Then they are saying that mutation. When I was explaining this, I called it a devil, that because many students, they don't understand it. Yeah, sir. Someone say that sir bonge okay ntando say 1 point. If I said let's bro ridge, will they cross it? Let's bro reach 1.6, 1.63. I don't know. Let me check. 1.63. Does it make it to be binocular? Because the question was about binocular. Does it make it to be binocular? I don't know really, but they might. But you are looking for something which make that organism to be what? To be binocular. You understand? Yes. Then we go to the next question. Explain. This one I explained it several times, and I say that you want it or not, that question is gonna come because I knew that it's gonna come. How did I know that it's gonna come? Don't ask me, because I'm a teacher who has taught for many years, and I know that. You know what happens when we see that you fail something? We keep bringing it until you understand it. If you don't understand it, we will bring it, we bring it, we bring it until we see that you understand it. That's why we bring things which you see that you have failed them. We bring them again. Is it fair? I don't know. Do they want you to pass? So you want all of you to pass? Who's gonna give you jobs? I'm just asking myself. Explain the effect. They want to know the effect this mutation has on the protein. The effect this mutation have on the protein. So what happened and how do you write to get these 4 marks? Saying that mutation caused triplet 1. So let's look at triplet 1. Triplet 1 is TG, TGT. Me, I use the TTT. Okay. Changed to TGT to triplet number 3, which is TGC. TGC, isn't it? So now I need to know what changed. This and this. Meaning that this was changed. So you write in exam that TGT, the third, you can say that the third base T was changed to C. That is the first mark. As I told you that TGT is changed to this. So you need to tell us what changed. The third base T was changed to C. That's 1 mark. I'm not saying that that's how they're gonna give you a mark, but that's how it's supposed to be. Yes, so it depends on that, but that's 1 mark, obviously. The second bullet will be you change this and you tell us which amino acid do they code for. The second bullet, this TGT is gonna be ACG. A. And then this one is going to be, when you change it to messenger, it's going to be A, C, S, C, G, isn't it? Yeah. Then now, SCA code for what? So this is another mark, guys. And then the first one, when you are writing it, this is one mark. This one mark. Hope, yes, all of them. Then you tell us, what do these ones code for? How did this one code for? ACA, ACA, AC, SCA is here, which is threonine. And then SCG, SCG, SCG is the same thing, threonine. So meaning that now you tell us that these two code for the same amino acid, which is threonine. You understand? And then because the same amino acid, so it's another mark. Because the same amino acid, therefore there will be no effect on the protein. No, that word no effect on a protein is also another mark. So another one mark, change this one. The third base changed from this to this, one mark. Then you tell us that this code for this, so you, another mark. And then you tell us that this code for threonine, and this one also code for threonine, another mark. And then there is no change in the protein, that's another mark, which are 4 marks. That's exactly what I said in a paper or in our preparation, and then I hope you got them right. Thank you, my boys. Thank you, my girls. Right. Let's continue to the next question. Let's continue to the next question. 2.4. 2.2 diagram below shows the cellular phase of meiosis, a phenomena that can occur during this phase of meiosis, which may lead to chromosomal mutation. All right. At the end of meiosis, gametes are produced. Cool. Explain one other biological importance of meiosis. So here you talk about variation. They are talking about biological. You talk about variation. You talk about maintaining the chromosome number from generation to generation, because production of haploid gametes, they talked about it. Tabulate the difference between meiosis 1 and meiosis 2. I told you, go phase 1, go to phase meiosis. Prophase, crossing over takes place. There's no crossing over in prophase 2. You go to metaphase, homologous chromosome align along the equator in pairs. Then other side, chromosomes align along the equator singly. Then the next part, you talk about chromosome, homologous chromosome move to opposite poles. The other side, chromatids move to opposite poles. You form 4 cells. So here you talk about all the differences, the way I showed you in the book. So I don't need to go through them. About them like many times. You understand? Yes. So that is it. Then we go to the next question. Yes, these are the differences we said you must write about. There are the differences. Then we go to the next question. What are they saying? What are they saying? What are they saying? The next question, what are they saying? So the table must be a table. You draw a proper table, like, then you put here meiosis 1, meiosis 2. Bullet number 1, you write. Bullet number, you write. Separate the line. Bullet number 2, you write. You separate the line. Bullet number 3, you write. Yeah, it must be a table. It must look neat. Don't just draw a T. Hi. Then they're saying that name and describe the phenomena that occurred in this. The name, you see, they said name. So in this case, the phenomena is non-disjunction. So that is one mark. And then how do you describe it? They're not saying explain. They're saying failure of the chromosome to separate. Yes, or these chromosomes failed to separate, this chromosome failed to separate, and then the whole pair is pulled to opposite, it is pulled to one pole, you see. So it's being pulled to one pole, and then this was mitosis was anaphase 1, you see. It is anaphase 1. I don't know whether they mentioned it. If they didn't mention it, then could be another mark. So at the end, yes, at the end of meiosis in the cell, if the gamete containing 3 chromosomes fertilize the gamete containing 2 chromosomes, would this lead to Down syndrome? I bet that many people, guys, please don't tell me that you said yes, because I emphasized it. I emphasize this, and I say that I'm emphasizing it so that you don't make the second mistake again. I'd even left, and then I said, oh guys, I remember this. So remember, at the end of meiosis, let's read again. If one gamete containing 3, if one gamete containing 3 chromosomes, I see, fertilizes a gamete containing 2 chromosomes, would this lead to Down syndrome? That's the answer. I will say no. You know why? This is not a human cell. Did I mention here the human cell? Never. And when I told you, what do you see here? What do you see here? How many chromosomes do you see? Do you see 46? Do you see 23 pairs? No. You see how many? I see 1, 2, 3, 4. So if one cell has this 1, 2, 3, you understand? So does it lead to Down syndrome? The answer is no. Why? Because this is not a human cell. It's not a human cell. Yes. And they didn't mention that this is chromosome number 21. It only applies to chromosome number 21. Do you understand? Even when we are talking about it here, when we are talking about it here, check. They say that 3 number chromosome 21. And here, even here, they don't say 3. They say 23 plus 22 gives you 45. Or 23 plus 24 gives you 47. They don't say 3, they don't say 2. Unless they say 3 for chromosome number 21. You see that? With the 3 number 21. You see? Number 21. You see? With the 3, it must be specific to chromosome 21 for someone to get what you call Down syndrome. Please don't repeat this in the final exam. You understand? And I emphasize this, that if you don't see human cell, please don't talk about that. Yes. So I think I'm done with that. Then they're saying that, what is happening with me? Safia. Thank you so much. I don't know what's happening with me. Maybe I'm so excited and stuff like this. Yes, he's saying that this I didn't. Thank you so much, Safia. Draw a diagram of a cell in a phase before. That's exactly what I say. That, hey, as if these people, they are reading my mind. Anyway, draw the phase of this cell before one that's shown above. One shown above is anaphase 1, because these are still full chromosomes, you see. So the cell before you draw for metaphase. But now they said labeled. Make sure that you label everything. And then number 2, make sure that you give a caption. Title. It must have a title, 1 mark. So these 5 marks is for Mahala. Honestly speaking, however much it had some challenges, but they brought a lot of open questions for you to answer and get marks. Yes. Let's go to question 2.3. This one, I didn't go through it. That's what I said. And I say that everyone knows about it. All right. Although a rare occurrence, Cases of babies using switched birth in the hospitals have made new. Okay, you guys, you read this. Since the individual sequence, okay, unique. So they used restriction fragment length polymorphism. Yes, this is the first one. It's the method. Or they're using polymerase chain reaction, which we call the PCR. They call it is a PCR machine. It's a very small thing, but it runs, spins very fast, very fast. The PCR machine, yes, and very expensive. Yes. Below are the results. Okay, the DNA. They are saying now question number 1 from the text. State 2 methods they used to analyze. So these are free marks. So they say they used this and then they used this. They are saying beside on the DNA profile provided, which baby or babies was currently allocated by the hospital. So in this case, correctly allocated by hospital. So what you need to do is you need to go this, to go this, to go this. Like you have to match them using a ruler. Yeah, so you have to use a ruler and then you see that these things are matching. So after I did my matching on the screen here, so I saw that I saw that baby number 2, was correct with the couple number 2. So it means that, so if you, this is a father, no, okay, baby number 2, they said baby number 2, baby number 1 was given couple A, baby number 2 to couple C, meaning that, yeah, this one, okay, let's find out. This matches with that from mother. All of them must be matching. Yes. And this one matches with the mother. This one matches with the father. If the mother, this one matches with the mother. This one matches with the father. You see that all of them are finished. There must be no bar which is left, which is not matching with either father or mother. So 2 becomes the most correct answer. Yeah, I hope you matched correctly. Yeah, if you find another one, try to match. Yeah, but based on what I've matched here with my eyes, but you're supposed to use a ruler, you understand, so that you bring it correctly, you understand. But since 1 mark, maybe it's that is. Explain your answer. The answer is explaining that you see all these bars. Yes, they match with the father and mother, you understand, all of them. Or the bars which match with the mother, the remaining bars must match with the father. You see? Yes. Of the remaining bars of the kid must match with the father. Or those which match with the mother, the remaining bars of the kid must match with the father. So that is it. Provide one other use of DNA profiling. I told you, go read about it. You have a lot about this. Talking about donor, talking about, what happened to my system now? Yeah, talking about donor, talking about the forensist. Yes, how do they use it? We can use it to identify criminals. You understand? So this one is 1 point. If you say criminal identification by, you understand? Yes. Diagnosis of inherited disorder. You understand? Yes. So you give those answers. Then we go to the next question. Someone saying 2.23. 2.2. What's happening to me? Wait. 2.2. 2.23. Oh, yeah. I answered it, guys. I hope I answered it. Yes, I answered it. The one which was talking about the diagram. Yes, I answered it. Hey guys, I hope I'm not. Yes, I don't know. I don't know what to say. All right, let's continue. So I think I'm done. I've not skipped any question. Eh? If I've skipped, guys, let me know. Eh? 2.4. 4. Yes, yeah. They're saying that in this species of fly, Drosophila gaster, yes, which we call the fruit flies, yes, tan light brown, body color is basically dominant and dominant over black. So this one color is dominant over this, and then they're saying that long, long, this long, meaning that this one is capital T and long is dominant over this is small l, and then this is small t and l. Okay, sharp. The pinot diagram below shows the cross between the female and the male. Okay, Drosophila melanogaster fly as shown. State why the type of cross involved in this scenario is referred to as a dihybrid cross because two characteristics were involved. So you can talk about them, the color of the body, and then you can talk about the length of the wing. Yes. Then they're saying that state, okay, the formation of gamete with different allele combination can be explained by Mendel's laws. Name two laws that apply. So the formation of gamete with different allele combination can be explained by Mendel's laws. Name two laws. Mendel's law of independent assortment or Mendel's law of segregation. And then number two, you also have Mendel's law of dominance because one is dominant over one is dominant over the other. So Mendel's law of dominancy and this Mendel's law of independent assortment or Mendel's law of segregation. Yes. They're saying explain your answer. Oh, so basically, where there is a capital letter or a recessive allele, the dominant allele will only express itself. You understand? And then when during assortment, during segregation, these alleles will separate independently. The capital T will not influence small t, and the small t will not influence the capital T. They will segregate independently. You see, they will segregate independently. You see, so that's how Mendel also that there is no influence of one allele by another. You understand? Allele in another locus. You see? Yes, because these alleles are going to be going to different loci. So they're saying that identify the genotype of gamete of male A, the genotype of gamete. So this one, they're looking for that. How did I form this? Look at what is formed. So it means that if the capital T came from here, small t came from there. So it's going to be small t. And if capital L came from there, small l came from here. So it's going to be small t, small l. So that is it. It must not have space. Then the genotype of offspring B. Genotype of this. So now since I know this, capital small t, small l. So I'm going to say capital T, small t, small t. So it's going to be small t, small t. And then it's going to be small l, capital L. So that is Mendel's law of dominancy. So meaning that if the dominant allele is there, a recessive allele can't express. And then you start with the capital letter. So that is the genotype for individual B. They want the phenotype, not the genotype. Ne? Since they want the phenotype, then it means that it's gonna be small letter is what? Small letter is gonna be black and long because capital letter is there, the dominance is there. Long wings, black and long wings. Then the genotype of a female parent, genotype of the female parent, genotype of the female parent. Where is the female parent? So we see that Yes. So we see that this one, they say that, what did they say here? Did they say, uh, based on what we see here, we see that these were heterozygous. This is heterozygous. You understand? This is heterozygous. No, 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 no. Where it came from, né? Because these two they combined, so it was like this: capital letter L, capital letter small t, capital letter L, and then capital letter L. Remember, this was they say that white and long. So that is capital letter T, small letter t, capital letter T, small letter t. So that is it. And then capital letter L, small capital letter L, and then it gives that. But because it is a genotype, we don't circle them and we don't leave space. So it is capital T, small t, capital L, like that. Yes. That's it. I hear. Yes. So I think clear there. Mm, yes. Someone was asking a question that. Yes, sir. Hi, sir. I'm seeing your memo and I'm super stressed. My hair might follow. Don't please. Don't please. Don't please. Don't do that. Don't do that. Don't please. Ne? This is just a guide so that your hair doesn't fall off. Because now you still have a chance to modify your life. You understand? Yes, you still have a big chance. And I know that when you experience that and they bring the same thing, you will never fail that again. I'm telling you, you will never fail that again. So we have explained the Mendel's law of dominance and the Mendel's law of independent assortment, or Mendel's law of segregation. Yes. So I think I'm done with those ones. Let's go to question 2.5. The diagram below shows two models of evolution based on the rate at which it occurs. Yes. I talked about this punctuated equilibrium and gradualism, guys. Ish. And I emphasize and say that please don't make a mistake. And I know that there are some people who made mistakes and they went without having it. You understand? And I emphasize that please, okay, just help me go to the paper. Please don't go to the paper without knowing this, without being explaining that. Therefore, there we go. And I told you that if you see something with the ladders, with the ladders, that is punctuated equilibrium. That's what I said. And you see that something has no intermediate. There are no intermediates here. That's punctuated equilibrium. You see something is continuing gradually. That is gradualism. And even used an example of people who go to ShopRite and buy Gentle Magic. They start to change their skin slowly. Ne? It doesn't today here, tomorrow. No, it takes weeks so that the skin keeps on changing. You become bright and bright and bright. So I think it's clear. Next time you won't fail it if you fail it. If you got it by chance, then it's cool, still fine, cool. And then tomorrow, make sure that you're able to explain it. Give the letter of A or B. That would be best present gradualism, obviously. So gradualism is gonna be B. And then this guy explained the evolution. Name the letter A or B. Yeah, this is punctuated equilibrium. Yes, punctuated equilibrium, you see. That's the guy. Punctuated equilibrium. They brought him here. Exactly. Yes, so it is a punctuated equilibrium, so the answer becomes A. Name and describe the model of evolution in A. Name and describe the model of evolution in A. Where is the model of evolution? They are looking at this. They're saying to describe. So when you say describe, name, meaning that punctuated equilibrium, you have uh, 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 um, you have explained. That's why they used this his name. You understand his name, so that when they ask you here, they ask you what is it. You understand? So it is punctuated equilibrium. So your answer was supposed to be punctuated equilibrium, the way how it is here. And then now you come back and say, describe the mode of evolution. So here it has long periods of no change with the rapid periods of what? Change. Did I say the rapid? Yeah, long periods of no change with the rapid period of with the short period of rapid change. Long period of no change with short periods of rapid change. Long periods of no change with the short periods of rapid change. Longer periods of no change with the rapid periods of with short periods of rapid change. So that's what they are trying to ask you to explain. It is made up of long periods of no change with the short periods of rapid checking add, because you have already identified, so 1 mark is already gone. Yeah, name and describe. Name the type of fossil that support the model of evolution in B. So in B, that is the gradualism. Basically here we use fossil records, the what, the skulls and stuff. So we have different, basically we can talk about the body fossils, because the types of fossils, I know there are body fossils, molecular fossils, we have trace fossils, we have carbon fossils, we have pseudo, we call them pseudo fossils. They are not fossils, but they look like fossils. But in this case, we use those remains which are we can able to see, like the skulls. When you talk about how did they evolve, how do they form these transitional fossils, né? Yes, that's how you explain it. So the type of fossils we see, you see that it has these transitional fossils, né? Yes. So basically it explains everything about the body fossils involving transitional fossils. Why? Because here we don't have those transitional. Here in punctuated equilibrium, we don't have those transitional fossils. So yeah, I hope they will give all them, or it will depend on what they have discussed, or based on your level. Question 3. I hope you guys, you got 50 out of 50, or at least 40. Last time you got 40 out 45, and then now 40. So that is the 85 out of 150. So now let's go hunt for some other 40 marks or 35 marks, and then we see if we can get it. Maize. Domesticated crop have been transformed over thousands of years and now look vastly different in comparison to their relatives in the wild. So they're saying that the maize belong to genus Zeme, which includes wild Antios Ind. This is scientific. They know them as Zime. Yes, Zimes. Okay. Okay. So they're saying that this produce cob only 6 to 12 in rows, protected by the hard outer covering. Modern maize produced by farmers have cobs consist of 500 or more exposed to. Yeah, I didn't know. I'm just eating meal. Yum, 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 yum. Even I did. Oh, who knew how many lines do you have on the maize? We are just eating, enjoying, putting salt, and yeah, we throw the cob. My God. Diagram below shows the wild. Yes. Name the process through which human produced. This is how do you call this process? We call it what? Artificial selection or selective breeding. This is it. This is the maize they are talking about. Here we go. These people, they will kill me, ne? Yeah. So this is the maize. They brought this maize and they brought this maize. That's it. That's it. So it is artificial selection, ne? Yeah, sometimes you call it selective breeding. All right. State one negative effect that process mentioned above may have on the species. So first of all, the one which are not desirable might get extinct because they're not given a chance to reproduce. That's number one. Number two, it might change the taste of these individuals. You understand? Yes. Number three, you can talk how many they want. They want only one. Describe how this. Yes. Yeah. Number three. Okay, you can also mention any of what you think. So, but we humans start to influence. The nature cannot determine what is the product. You understand? So, meaning that there is no natural selection there. And if there is no natural selection, when you're just going with what you want, some genes of this species might disappear completely. You understand? Yes, because it's not about the best suit. It's about the desire for the human to produce that. Describe the process mentioned above. It was carried out. So basically, you just explain this. Just explain this. So what did we want? What do we want in this regard? Yeah, yes, you just explain this. Yeah, that choose or select the maize in the field. You understand? That produce more maize, more this. You understand? So what happens? You see, you modify them, and then now let only those maize reproduce because they have more of the rows of this meal. Then select the offspring that produce more on this cob. You understand? And let only those offspring reproduce, meaning that those maize which have more maize on the cob are the one which are, you understand, allowed to reproduce. How do you do cross pollination, or you allow those ones to pollinate each other? You understand? And then you keep repeating this until this big maize is obtained. This is not just one day, it's about many generations. Yes. So describe how this product. Okay, we are done with that. Explain one economic benefit of producing modern maize cob has to the farmer. Yes, the modern maize cob, it brings about more food availability, and then they can sell, they get money. You understand? Yes. So basically that's it. Number two, you can also talk about increase the resistance, but is that economic? Yes. If it can resist the drought, then automatically it means that the person will get more money. If you can resist the diseases, then automatically it means that the farmer will not use more pesticide, insecticide. You see? Yes. So more cob, like more on the cob, meaning that the farmer will be able to sell and then get a lot of what? A lot of profits. So any of one, then which you can explain, then you were supposed to get a mark in that regard. Yeah. Let's finish up. Someone saying, why me? Okay, they're saying that I've been getting distinction because of you. Thank you. I think I might go to the national course. My average is 95%. Yeah. Let's clap for you. Yeah. Yes, man. So I got 150 in this paper because of you. Oh my God. Thank you so much, guys. Thank you so much, guys. Hey, you make me proud and make me not sleep so that I prepare you more so that you maintain this. I feel Oh, last time, I feel so good. I feel so good to hear that. And guys, those who are still low, you can still make it. I'm telling you, you can still make it, and you can still improve your marks in this remaining time. Even if it's day, still you can make a change. I'm telling you. Yes, let's continue. Okay. Let's go to the next one. They are saying that. Uh-huh. Okay, connected to two large mess. Okay, this two large mess. You guys, did you read this? It says, and is separated by two water bodies. Saying that this island of Panama is narrow strip. Okay, strip of land that joins also. So is it? I think it is this. Oh, it's this, ne? Okay. Let's say North South America. Some believe that this strip of land hmm, formed 3.5 million years ago. The Caribbean Sea is warm. Where's the Caribbean? Oh, this is the is warm. Is warm while the Pacific Sea is cooler. This one, okay. Then they're saying that pork fish. Muslims, do you eat this one? Pork fish. Maybe it looks like a pork. I don't know. Or tastes like a pork. I don't know. Okay, can be found either side of the narrow strip. Okay. However, when scientists placed the male porkfish from the Caribbean Sea, okay, ocean into the same fish tank, they would not cooperate. They are now considered to be all right. So I have porkfish from Caribbean, and then porkfish Panamic. Panamic, we have the Panamic and the Panamic porkfish, and then you have the porkfish. Okay. Define species. Species, that one you know. Ne? Group of organisms that can interbreed to produce fertile offspring. I said yesterday that fertile offspring, not viable. Name two isolation mechanism. These are the one. I said that I'm not going to go through them, but you have to know them. Yes, this one. See? Say don't go without knowing them. Name two reproductive isolation mechanisms that have been prevented from okay, porkfish from copulating. What do you think? What made them to not to copulate? First of all, it could be the distance. You understand? Yes, it could be the distance. Basically, is temporal what? Or the distance between the two. Then number two, because this one is warmer and this one is cooler, you understand, so maybe they are breeding at different seasons of the what, seasons of the year, so it also could be a problem. Yes, so there is a barrier prevents them also from having this gene flow. So, based on the scenario above, describe speciation of porkfish through geographical isolation. Now, how are I supposed to explain this? Today they put it 7 marks. They usually put 6 marks. As I said it, make a mistake to go to the paper without knowing this. And if you did that and you know that what is the results. Okay, let's finalize it. Let's finalize it. Let's finalize it. Let's finalize it. They're saying that there was. Let's find out who was the ancestor. Okay, large two masses separated two. Panama. Okay, Bob. So basically, there was one group of these porkfish, né? Isn't it? Yes. And then this it was separated by what? By a geographical barrier, and then you have to tell us which geographical barrier was that. This canal, this canal, you understand? Separated into two different populations. You understand? There was no gene flow between this and this. because they are separated by different geographical barrier, so they experience different environmental conditions. You understand? Different environmental conditions. So natural selection occurs independently. So it was one group of porkfish. Then what happened? It was separated by a geographical barrier, which is this. You have to mention it. Yes. Form two different groups of porkfish. And then now they experienced, there was no gene flow between these two porkfish. You understand? Because of this barrier. And then natural selection occurred independently because of the different now environment. One is cooler, one is warmer, you see. And then what happened after that? Since natural selection occurs independently, then due to the different environmental conditions, then they became genotypically. You start with the word genotypically, and then phenotypically different. You understand? And then when you bring together these porkfish, they say that they cannot, if you remove the barrier and you make them mix again, they cannot interbreed to produce fertile offspring. They said it here. They cannot. They cannot interbreed to produce fertile offspring. They said it here. They are now considered to be two different species of this pork fish. Then you can maybe mention it, i.e., this name, and this name. You understand? Yes. Yeah. Sometimes those two names could be a compulsory mark that if you don't have it, you have not written it, out of 7, you're gonna get 6, because they wanted to see these new species which were formed. You understand? Yes. Lastly, but not the least, is the pedigree. Okay, saying skin disorder caused by chromosome. You see, I told you that it's sex chromosome. Ne? Is it sex linked or non-sex linked? So we know that it's sex linked. So now you need to also know whether it is dominant allele or recessive allele. They showed you here it is a recessive allele. Now that's cool. Why do I know that's recessive allele? Because they said individual B. Individual B here, individual B, they said affected male, meaning that is caused by a recessive allele. Name the type of diagram indicated by pedigree diagram. Provide a possible genotype of individual A. Provide a possible genotype of individual A. Individual A, if you look at individual A, produced a baby who is normal. So it means that the individual A, since this one is normal, so, and this is sick, then automatically individual A could be XX, capital T, yes, even X is X small t Y. So if this combine with this is gonna form X capital T, X small t. So this individual is still female, but is still normal. You see, it's cool. When it's like this, there is a chance that this combine with still this to form still this. So I think this, they will give you a mark, and they give a mark. How many marks we are given? 2 marks. Yeah, cool. That's why they say genotype or genotypes, so that you write these two together. Then they're saying that explain the relationship between inherited genotype and phenotype of individual C and the Mendel's law of dominancy. They wanted the Mendel's law of dominance. Yeah. Mendel's law. Today they brought all the Mendel's laws. Explain the relationship between the inherited genotype, yes, genotype, and the Mendel's law of individual C. Let's find out who is individual C. Individual C, hey, yeah man. Individual C is here. So if it is individual C, explain the relationship between inherited genotype and the phenotype of individual C and the Mendel's law of dominancy. Individual C, where are you? Okay, individual C, we have seen that. Oh, individual C, first of all, it is a female who is unaffected. So that is the phenotype. But the genotype, the genotype, the genotype one, this individual C is like this, capital letter T and then small letter t. Why? Because this father gave that individual a small letter. So now how are you going to explain? So basically this is a genotype. That's mark 1. Then you tell us the phenotype. It is normal. It's unaffected. You understand? That is another mark. Then the other 2 marks, you tell us that because you need to talk about the inheritance. Yes, obtain this dominant allele from the mother and obtain the recessive allele from the father. Yes. So basically that's what they wanted in that regard. So explain why males are more likely to be affected by disorder than the females. So we explain this also, that males have one X chromosome. If a recessive allele of T appears on it, the individual is having the disorder, is affected with this disorder. Yes, while female has 2X. If a recessive allele appears on one. Don't know why they give it 2 marks, because it's supposed to be around 3 or 4, whatever. Use the genetic cross, show how individual D inherited. Individual D, again, they're asking the same thing. Inherited the genotype which caused this individual to be affected by the disorder. To be affected by genotype in the correct position on the right. Circle. Okay, they're saying circle the genotype in the correct position on this cross. Use the genetic cross, show the individual how individual D, okay, that one is cool, inherited the genotype. Okay, individual D obviously is a male, whereby is having X, small t, and a Y. So since this one is heterozygous, then we can. But they say using the genetic cross. You know what is the first point? As I told you, that the first point in this case, you're gonna write this individual D. Ne? So this individual D was on the second generation. So you're gonna say P2 F2, because P1 is this. Ne? Then this is P2. You understand? Second generation. So P2 F2. Then you say phenotype. Yes. And then here, okay, let me just do it on the blank page. Hey, someone saying that, yo, hi guys, wanna hire from Cape Town. How many marks does evolution weigh? Oh, all right. Yeah, they're saying, let me just use this blank page so that you're able to understand what I'm saying. So we are answering this question. Ne? They're saying use the genetic cross to show how the genotype D. So genotype D is this one. So how did this individual inherited? Caused by individual to affect by the disorder. Okay, so basically what happened is I'm gonna have P2, okay, F2. Yes, phenotype, phenotype. Then I'm gonna have genotype, genotype. Okay. Then here it's, I think this one, remember now these are the parents. So it's gonna be normal. So it's gonna be normal female crossed with normal female crossed with normal male. Okay, sharp. Then the genotype, when we see it here, we say that this is a heterozygous, and then this is normal. So it's gonna be X small t. It's gonna be X capital T and X small t crossed with normal male, which is X capital T Y. Then you say meiosis. Then when meiosis occur, so you're gonna have capital T Let me increase the thickness, capital T. Then here is gonna be then it's gonna be capital X, small t. Then this one's gonna be X, capital T, Y. You see? Then these are gametes. So let me just make this one far. F2. So now I have to cross this, whereby this is gonna go with that. So I'm gonna have X, capital T, X, capital T. I'm gonna cross now this with that, whereby it's gonna be X, capital T, Y. And I'm gonna cross that with that, which is gonna be X small t, X. Okay, let me start with X capital T, X capital T, X small t. Then now this with that, which is X capital T, Y. So now this one no, this one no, this one no. Oh, did I cross it properly? Okay, that's why I prefer using a Punnett square. Okay, this with this. Capital T, capital T. This one is correct. This with this, you see? Capital T, Y. Then now, this with this. Okay? Yes, it's capital T, small t. Now this with this is small t. This is a small t. So let me just say this one. So they said circle. This is the one which caused this situation in this boy. Yes, this is the one. They're saying that circle the genotype in the correct position of the cross. You know what they are saying there? Yes, this is the one. So it's basically like this. That's the one which caused that individual to be like that. How many marks are there? Then you put you can put phenotype, genotypes, everything, so that if any mark is there, they can still give you. I told you that when it comes to this genetic, you will write everything Kaufera. No one is gonna cross you because you overwrote. Yes. The last question is Nala and Luke are farmers of the same species of fish, small fish. Nala breeds homozygous yellow fish, while Luke breeds homozygous blue, and then this is yellow. Okay. Ooh, the question is going. In the investigation, determine the dominant color in this species. They allowed the investigation determined the dominant color in the species of fish. They allowed for yellow fish to breed only with the blue fish over 5 years. Okay, over 5. Oh, this is a key point. And collect 100 species each year. This is another key point. They say they allowed yellow and blue only. So it's another key point. Okay. Name the type of dominancy which is indicated there. Okay, let me check and see. It's this. Yellow and blue. Why is this? So this one becomes an intermediate. Therefore, it is incomplete dominancy, whereby you have an intermediate. It's not yellow, it's not blue. It's just a medium color, which is an intermediate. Therefore, it is incomplete dominancy. Incomplete dominancy. Provide a reason for your answer because you produced a new character or an intermediate color of the offspring. Okay. Name 2 ways in which the the reliability of this investigation was ensured. Hey guys, here we mark the English. As I told you that here we mark the English. You need to know which English do you use. Name two ways in which reliability. Reliability is done by improving by repeating the experiment many times. So how did they do the reliability here? They repeated the experiment over 5 years. So that is the key point. They repeat the experiment over five uh, five 5 years. You see, that's number one. Number two, they used a large sample size. They collected 100 offsprings each year. You see, 100 offsprings each year. You understand? 100 offsprings each year. So this is the first point. This is the first point. This is the first point, and then this is the second point for the reliability. Give one way in which the results of this were made more valid. How did they make this species? How did they make this experiment to be more valid? Here we use what did they use? They use the same. They only allowed the species. You see, they only allowed these species. You see, to the one yellow and the blue to mate. You see, yes. So they, meaning that they kept the same type of species. Yes, they only used the same. The blue were supposed to be heterozygous, homozygous, and also this one is supposed to be. So that is, they use this. What did they use? Same way. They didn't change it over the experiment. That's what they are. So if you wrote any which was used, so they will credit you hopefully. So give two planning stage. So planning stage, you see that they had to identify the homozygous blue species and also homozygous, you see. Then number two, where did they fish them? They had to prepare where these fish are going to be bred from, like maybe the pond and stuff, you see. So how did they obtain them? Maybe also the workers who were supposed to be there, they had to train them. So how do they do the planning? Maybe is it allowed to just do the fishing without permission? So what did they do before they did the experiment? What did they put in place before the experiment was done? So bar graph represents this one. Obviously, you know, this is supposed to be on the x-axis, this is supposed to be on y-axis. And then remember, when you're marking the bar graph, it's supposed to be spaces in between. Yes, spaces in between. This space must be the same as this. This bar size must be the same as this. If you don't do that, we give you 0. Caption must be there without units. Here it must be there with units. Here it must be there with units. And then this is what the figures go here. But if you start with 0, 2, 4, 6, the interval must be the same. Then now here you write the, what was it? The colors, the green, the blue, the yellow. Yes. So after that we come here, title, the type of graph, the caption, the axis. We give you those marks accordingly. But I don't expect anyone to fail or to fail to get those marks. According to Darwin's theory of evolution, there is a great deal of variation. Yes, according to Darwin's theory of evolution, there is a great deal of variation in offspring. Use the natural selection. Describe how variation of offspring increase the number of individual with the favorable characteristic in the next generation. So what are they looking at here? According to Darwin's theory, okay, there is a great deal of variation, you see. Using the natural selection, describe how variation of offsprings increase the number with the favorable characteristics. So basically, it's a generic question. There is no example they use. So they will tell us, you describe natural selection. Yes, it could be This is what they are looking for you to explain. This is what I wanted. This. Yes. Organisms produce large number of offsprings. There is a great deal of variation among these offsprings. Then they'll tell us that some have favorable characteristics, yes, for survival, while others do not. They don't have these favorable characteristics. So when there is environmental changes, those with favorable characteristics will survive, and those with unfavorable characteristics will die. You understand? And then the organisms that survive reproduce and pass on allele to their favorable allele to the next generation or to the offsprings. So that's what they are trying to. They wanted exactly without change in that. Yes. So I think, guys, yeah, if you wrote Mendel's law of assortment for God independent, I don't think it's wrong. So hopefully it's correct. Yeah, guys. Yeah, I think that this read the question. It says show how C inherited the disorder. It is supposed to start with the P1. Okay, Bong Tando, you're saying read how C inherited disorder. Okay, if you have a question, let me know. You are saying that Tando, you are saying that they said here, describe how C. Where is C? Oh, serious, Tando? Let me check. Tando, thank you. Let me check. We're saying that. Oh, explain the relationship between the genotype of C. They're not saying that. They're saying use the genetic clues. Show how individual D inherited. Tando. They're not saying, sir, read the question. It says that show how individual C inherited disorder. It is not supposed to be P1. Stando, they say that explain using genetic cross, show how individual D. They're not saying about individual C. Individual C was mentioned up here. Ne? Individual D inherited the genotype which caused this individual to be affected by the disorder. You see, they want to know how this individual D, this is the first generation. Individual D is the parents are in the second generation. Do you understand? Yes, the parents are in the second generation. But sometimes this P1, P2, sometimes they say those who wrote P1, they will give you, those who wrote P2, they will give you, depending on what they have discussed. But if we are using what I told you, that the KO, the method where when the examiner sees the answer, gets it right, P2, this is P2. Why this individual is on P2? Yes, because this is P1 F1, first filio generation. The first filio generation, if you use this individual, therefore they become parent number 2. The parent number 1, offspring number 1. So the offspring number 1, they become parent number 2 for offspring number 2. So now this one becomes parent 2, then this one becomes offspring number 2. That's why we say P2, F2, because they are looking for D, not C. If it was C, then it would have been correct. But they are saying individual D. I think I'm clear there. Yeah, if there is any other question, sir, okay. Thank you so much, Karabo. I think, guys, everyone is satisfied. I don't want to go away when